Hello, in this video we're going to look at the Nobel Prize in Physiology or Medicine in the year 1908 for Ehrlich, Mechnikov and Paul Ehrlich in recognition for their work on immunity, and we're going to look at their Nobel Prize lectures, two thus. First, Ehrlich Mechnikov on the present state of the question of immunity in infectious diseases. I've summarized this lecture. It's quite long. It's estimated that it took 1.5 to 2 hours when this lecture was given, so I've reduced it to a quarter, so some elements of course had to be skipped. It's mainly about phagocytosis. Here you see phagocytosis in action. You see that the enemy material is swallowed up by the white blood cell. Okay, here you see another macrophage which is swallowed up by the process of phagocytosis, and you see that the receptors, antibody, antigen have to fit. We come back to that later. Here you see another representation of that process. Okay, then the Nobel Prize lecture by Mechnikov, summarized by me and summarized quite severely. I estimated that it was already 9,000 words and I reduced it to about 1,500. So, of course, a lot of things had to go, but I tried to make it a logical story. Well, health of perfect interest. Here, pure research becomes practical. The human body is capable of resisting many harmful agents, but immunity is not the same for every individual. Pasteur found that immunity could be conferred by means of vaccination with microbes. Pasteur explained by terms of chemistry of the environment, microscopic organisms not being able to grow in an organism that does not give the substances what they need. But some microbes could live in animals. They were immune. Comparative pathology had to be called in. Some of the organism's elements play no role in digestion but can store foreign bodies, since the elements were once part of the digestive system. In lower animals, moving cells can close over foreign bodies, that's phagocytosis. It was now the task to show this also happens in higher animals. First it was done with the larvae of starfish. When splinters entered their bodies, the splinter was surrounded by layers of cells. It is spontaneous reaction of the cells without the help from blood vessels or nerves. This is the first stage of inflammation. Cure comes from the victory of the cells of the organism over the microbes from the outside. The next animal researched were water fleas, small crustaceans, subject to various diseases. Spores that invade the daphnae, causing a mobilization of cells around them. Cells that correspond to white corpuscles in human blood. Many great names in science doubted this theory, but I still continue to research. And he was proven right, of course. Higher organisms, including man, are more complex than little organisms. They cannot be investigated alive under a microscope. Thus, the research focuses on blood and organs, which is more difficult. But also, here, an invasion of microbes is answered by an accumulation of mobile cells, the white corpuscles of the blood, phagocytes. In the case of some diseases like anthrax, phagocytosis does not take place, leading to death. But animals vaccinated against anthrax do not die. The bacilli are seized by the phagocytes. The same is true for many other diseases, for instance, the plague. Weil has maintained that in the case of cholera, the microbes are faced by other means than phagocytes. His conclusion seems to be wrong. The phagocytes do destroy these bacilli. Other opponents claim that phagocytes can only absorb dead microbes killed by the blood plasma, but it is shown that phagocytes often contain live microbes. Still others say phagocytes themselves are hostile and therefore ejected as pus, but pus contains phagocytes that kill bacteria. So that's wrong. It's often said that the blood serum kills the bacilli. Certain cholera vibrios are indeed killed in this way, but this is shown to be an exception. Most bacilli cannot be killed in this way. As long as the phagocytes are intact, the immunity takes place within the phagocytes. Various intermediate views were formalized: immunity in plasma and in phagocyte, the plasma as the first line of defense, the phagocytes as the second. Just as there are two bacterial functions in that view, there are also two substances. The first would prepare the microbes but not kill, and the second would kill the microbes. Some scientists say that the substance in the blood is a secretion of the phagocytes, but it is becoming clear that this is not the case. My and Levaditis research has shown that phagocytes contain substances that act just like the complement in the humus, in blood and liquid. Phagocytes that have to work in a fluid without the complement die and the vibrios, the bacilli, remain intact. Washing, cooling, and maceration of the phagocyte destroys the complement. In the meantime, it has become clear that the working of the complement is purely chemical, while on the contrary, the phagocytes are alive. The complement works much longer in the humor than in the phagocytes, thus the phagocytes probably contain an anti-complement tool. Our plea is one of identity for the complements contained in the phagocyte and for those in the blood serum. It is not yet clear if there are other substances capable of destroying microbes. The work of Peterson and Gruber indicates that substances like this do indeed exist. It is also clear that the humor can only kill the weakest microbes. The bacterial action of the complement is linked to the amboceptors of Ehrlich. He will come later. These substances in the humor do not destroy but help the action of the complements. The amboceptors are phagocytic products, as shown by Waterman and Citron. The action of the amboceptor is not known in detail. They will kill only weak bacteria and not the majority. Microbes impregnated with amboceptors and the complement fall easy prey to the phagocyte. See the work of Wright and Neufeld. Humans are those relatively unimportant against infectious microbes, but some still defend the modifying take place by the amboceptor complement, also called opsocins or bacteriotropins. They only research microbe in glass tubes, not in real living bodies, which are very different circumstances indeed. In real conditions, the phagocytes showed that they are hypersensitive to external conditions. Parvou showed that phagocytes cannot perform their function well in certain conditions. The absorption of the bacteria helped by the absorptions is important, but only the beginning of the digestion of the microbes by the phagocytes. If the absorbed microbe does not die in the organism, is defensive to the infection. Some bacilli, like those of tetanus, can survive long inside phagocytes and are set free when the phagocyte dies. In most cases, it's lesions, that's the damages, which prevent the phagocytes absorbing and destroying the microbes. Bale showed that bacilli eject poisons, aggressins, against the phagocytes. Wasserman and Citron showed that the aggressins can hinder the phagocytes, but the microbes can absorb the phagocytes when the latter are in a state of reinforced by the phagocytes. The microbes can be absorbed by the phagocytes when the latter are in a state of reinforced activity. Bering showed that the phagocytes can neutralize poison that are produced by the microbes. Immunity can also be obtained by making the microbes inactive, after which they are killed finally. But phagocytosis is not secondary to this inactivation of the microbes. The phagocytosis also makes the poison harmless. See the work of Bezerka, Ball, and Weil. Thus, phagocytes ensure immunity from infections, microbes, and from poisons. Phagocytes can withstand many poisons. Bezerka selected the poisonic arsenic trisulfide, but the phagocytes dealt with it. Carl of Bordeaux demonstrated the same for lead salts. It's clear that the phagocytes are the main defense against dangers from infectious agents of all kinds. When microbes penetrate the white corpuscles, make use of the dilation of the blood vessels and the nerves that control it, in order to reach the battlefield as soon as possible. In immunity achieved by vaccination or an attack of disease, the organism modifies. The amboceptor excreted by the phagocytes prepare for phagocytosis. Thus, the phagocytes must also modify, but this is not proven yet. Peterson and later Salimbi brought phagocytes into organisms that were before in vaccinated animals, which gave real protection without the fluid being changed. Again, the property of the cells take pride over the humoral properties. It may seem paradoxical that the serious modifications that follow acquired immunity, the phagocytosis, show no augmentation of the function. See the work of Denis and Leclerc. At the same time, it was found that phagocytosis of an organism that has been under attack is different from that of an organism that has not been under attack. Maybe the strengthening is in the overproduction of substances produced by the phagocytes that prepare phagocytosis. There's discussion in the field about Ehrlich's amboceptors, for instance, by Baudet. The conflict about that is unresolved. Now to how all this is used in medicine. Says Choquet, don't know whether I pronounced it correct, first underlined that survival and recovery depends on phagocytosis absorbing bacteria. Vrijburg showed that the bacilli must also be destroyed, thus the extent of the phagocytation needs to be measured. Bam was able to use the degree of phagocytosis as a prognostic, as did Wright. In France, phagocytes are ejected to fight microbes. It was found that antiseptics poisoned the phagocytes, thus aseptics replaced antiseptics. Beer also used phagocytes to cure patients. Investigations have now started to strengthen the phagocytic reaction. Grunspan uses quinine, Neisser and Guerine studied a series of substances. It shows that we have made much progress in the last 40 years, as has done the theory of phagocyte in the last 25 years. Well, this is an extreme summation, I think, of the original text, which is at least 6 times longer. If you want to read the complete text, which is quite interesting and contains much more than I have just presented, then you can follow this link, which is also in the description under the video. You see, by the way, some phagocytosis of trophozoites and another more modern scanning electron micrograph of the process of phagocytosis by a phagocyte. You see the phagocyte is yellow, and anthrax bacilla, very dangerous bacilla, is the orange one. Okay, after this we will continue with the second Nobel Prize, which is by Paul Ehrlich on partial cell functions. Here we see 2 illustrations from the original lecture. Here you can see the link to it. We'll come back to these later, probably. And here you can see a picture of some trypanosoma parasites in the blood, which cause a terrible disease, of course. They will also be in the lecture, of course. Okay, I summarized this lecture 2. Is not as long as the other one. It's estimated about 45 to 60 minutes, so this is probably 3 times as short as that. Let's start. For a long time, the anatomy of the human body was the main field of research. This changed since the use of the microscope. Schwann established the cell as the final biological unit. Now the science of life revolves around this concept. Mainly dead and living tissue are examined. Now further penetration into the cell is needed, more than the microscope can do. The chemistry of the cells is what is next. Behring took a step in 1905, discovering the antitoxins. The toxins stimulate the production of antibodies. None of these toxins can be crystallized. They belong to the protein substances. I think they've now been crystallized, but for more than a century further. Toxins and poisons and stimulate the animal body to create antitoxins. Toxin can be deprived of the toxicity, so becoming toxoid, but retain their ability to produce antibodies. The toxoids still can neutralize the antibodies. The explanation: there must be 2 groups in the toxin, one of which remains in the toxoid and the other disappears. An antitoxin neutralizes only one toxin. They probably form a chemical bond in the neutralization. They fit each other, as Fischer says, like lock and key. Morground has proved that it is possible to break up the toxin-antitoxin again. The stable part of the toxin, heptofire, is the chemically active part. Toxics can adhere to animal cells and then cause a disease. The tetanus toxin, for instance, unites with the chemicals in the protoplasm of cells. I call these cells poison receptors. Wasserman has, of course, Ehrlich. Wasserman has proved This to be correct. The hepto-4 groupings must have a highly complex and stereochemical nature. So must the antitoxin, which is contained in the serum of immunized animals, thus must be discharge receptor. Excuse me. The poisons are not designed to be poisonous. By a coincidence, they disrupt the normal function of a chemical group they react with. Weigert stated as a general rule that if the deficiency is not compensated but made to excess, there is hyperregeneration. The cell gets rid of discharging them into the blood. Knorr stated that one part of toxin produces 1,000,000 times the amount of antitoxin. Some researchers think the process is much simpler. The toxin is destroyed, but the exciton is not. Repeating poisoning would accumulate protective power in the blood. When introduced to other organs, it can protect them too. See the work of Straub. He misses the point that an amount of toxin does not produce exactly the same amount of toxin, but 1,000,000 times more. We already saw that. The cell has a great number of separate functions. The toxin is a nutritive substance by accident attached to a lateral unstable grouping, which makes it toxic. It is proved that the hepto-4 non-poisonous and toxophore poisonous groups are completely independent. Remember that Krauss, Sistowicz, and Bordet proved that genuine protein substances can produce antibodies. Only the hepto-4 group is responsible for the formation of antibodies. The special relations between the hepto-4 antibody groups and the receptors is a vital important for serum diagnosis. Wasserman and Uhlenhuth revealed this relation for viral typhoid reaction. Immunization lures into the blood receptors from the cell that are found by the toxins. Examples of these receptors: preceptins, opposins, antitoxins, antiferments. It is now possible to study the link between toxins and antitoxins and the complicated reaction of the AMBO receptors. We already saw AMBO receptors in the previous lecture. I describe all the receptors which are designed to assimilate nutritive substances as nutriceptors. The nutriceptors are the source of the antibodies. There must be very many nutriceptors of different types. There are 3 types. In general, and this is a quote: 1. Those who do not enter the blood in the form of antibodies. It may be assumed that this will probably be the case with those nutriceptors which serve the very simplest function, for instance, the assimilation of simple fat substance or of types of sugar. 2. Those which enter the body in the form of antibodies mentioned and characterized above, and the development of which corresponds to hyperregeneration. Now 3. The third form presents a contrast to this insofar as it is now not a case of new formations, but a decrease in receptors. Experimental proof of this occurrence has, however, so far only been very rare. The only known instance is probably the evidence produced by H. Kossel that after prolonged immunization of rabbits with the hemotoxic eel serum, the blood corpuscles, as they did finally become insensitive to this agent, as though they had lost the specific receptors. Roll Gulbrandsen have also done work on the nature of this artificial loss of receptors. Frank Kleine and Mechnikov found that parasites can change so that they are no longer affected by the serum. The parasites that survived must have adapted. A biological change made them serum resistant. The serum must then also be adapted. Ehrlich describes this, calling the original nutriceptor A and the adapted one B. In adaptation, B thus replaces A. I say A, but it's A, of course. It has been produced intentionally and artificially, and it's hereditary. It is needed in how the triposome antibody influences the parasite. Now, like the trypanosome antibodies in more details. Usual trypanosomes have one grouping, for instance A, B, or C, unios, but there are also tryposomes with, for instance, 2, binios. If an antibody A acts upon a binio A and B, there is no damage. The same with antibody B. Only when they act both antibodies, there is damage. It again shows that A and B are nutriceptors. If amount of antiserum is very large, the parasite dies, but if the amount is not that large, the parasite may adapt. Ehrlich calls antibodies which have a purely anti-nutritive function attrapcells. The notion that there are chemical groupings in the cell for reception of nutrient substances is now proven by the presence of antibodies. More difficult is whether for the reception of left complication substances there are analogous functional groups. For oxygen and carbon dioxide, this is the case in the hemoglobin molecule with its iron residue. The protoplasm in red blood corpuscles does contain ferroreceptors. Off is that, of course. The proteins of red blood corpuscles does contain ferroreceptors. More difficult, is this also the case for different medicants? One cannot answer this now. Another question, do trypanosomes, the parasites, possess in their protoplasm definite groupings which govern the captivation of definite chemical substances? If a certain substance kills trypanosomes, an accumulation of it takes place in the parasite. I and Frank Browning and Rolls showed that one can have treatment against trypanosome strains that are resistant to fuchsine, azodine, trypan, etc. The acquired resistance is stable. The resistance is against a series of related substances. The trypanosomes that are resistant to all the three named substances are valuable in the search for a new type of trypanocidal agents. Another question was when to determine in what way the specific drug resistance comes about. Arsenic resistance, are toxoparasites, were investigated. Arsenic acid had no effect at all, being the most used therapy drug, in contrast to Koch, who said it would work inside the human body. Ehrlich found that arsenic acid in the body reduced to arsenious acid and cacodylic acid is reduced to cacodyl. The reduction is the reason why the parasites are killed by the new substance. Ehrlich in this lecture goes deeper in this with medical chemical details, which I have skipped here because otherwise it would become too long. But if you want to read it, go to the original lecture. It all tallies well with that carbon monoxide and other unsaturated compounds are toxic than their corresponding saturated radicals. The arsenoreceptor of the cells only take up arsenic residue, which is unsaturated, eager to adhere. Now we could examine the toxal strains in the test tube. The resistant strain needed a much higher dose to be killed than the normal strain. The minimum doses that killed the normal strain did not affect the resistant strain. The arsenoreceptor does in fact represent a certain function, the avidity of which can be decreased through immunization. There have been reached three different stages of relationship. Ehrlich presents these stages, but again I have skipped it here for reasons of length. Going toward conclusions. Proving is that chemoreceptors are connected with the constitution of protoplasm and undergo alterations if we alter the constitution of the protoplasm by mutation. The reverse, nociceptor being changed by inferring the chemoreceptors, has not been done in 1980. There are still gaps in the work, but there are many studies done in the field. But in general, we are getting into grip with the problem of obtaining an insight into the nature of action of therapeutic substances. If we follow this up systematically, we may be able to develop a rational drug synthesis and achieve complete cures. As again, quite summarized by me, at least cut into 1/3 of the original length. So some things are missing, some things I said I have skipped, but others sometimes I did not say it. If you want to read the complete lecture with all the argumentation, then you have to follow these links, which will also be in the description of the video. Here you see another illustration about the trypanosomatides, these play so an important role in the lecture. You see there are different variations. And here you see some drawings made by Paul Ehrlich himself, illustrating his side chain theory. And this is another one. If you want to read about the side chain theory, you probably better go to the previous video or, for instance, to Wikipedia. Well, for now, thank you for watching. Next week we will go into the Nobel Prize in Physiology or Medicine for the year 1909 with the general information, and the week after that with the Nobel Prize lectures. That's the scheme we follow the whole time. For now, thank you for watching, and maybe till another time.